Hello friends, in continuation to our previous classes, today we are going to discuss about fertilization events. The response to human being we are going to discuss. For convenience, we will bifurcate the class into two subclasses, okay, for better understanding. We have discussed in our previous classes that in spermatogenesis, that means in male human being, 1.5 million sperm will be produced per minute, whereas in case of female, just one oocyte per menstrual cycle, wherein the same will be arrested at second metaphase, that means of meiosis 2. That means it will be waiting for the sperm, that means sperm penetration, so as to complete the meiosis 2 and resulting to a larger ovum or egg and the second polar body. After mating, the sperm will be ejaculated into the female reproductive tract. Then followed by a sequence of events that ultimately leads to fertilization. That means of ovum or egg. That leads to zygote formation. Therefore, fertilization is a chain of events that culminates to zygote formation. For convenience, as we have discussed, we will be discussing about fertilization process in human being. The first step of fertilization is, approximation of gametes. With respect to human being, now we are discussing about here approximation of gamete in the sense the sperm, that means the male gamete which is motile, has to reach the female gamete which is a non-motile, and it will be taking place through a reproductive tract in females. It is with respect to internal fertilization. If it is external fertilization, the tract will be in water. That means approximation of gamete in the sense the male gamete should locate the female gamete is approximation of gamete and is the first step in fertilization. Number 2, after approximation of gamete, the second is chemotaxis. Chemotaxis means it is a chemical, the word implied, the chemical attraction of male gametes towards the female gamete. Sperm is a very tiny body when compared to egg, which is a relatively larger in size. Just like the sun and the earth, sperm is very tiny body. Millions of sperms in an average healthy human being will be ejaculated into the female reproductive tract, and only one sperm, what we call leader sperm, will be reaching the egg. That ultimately leads to fertilization. Okay. The other thing is chemotaxis. The major significance of chemotaxis is it is species specific. To avoid the fusion between that means a different species sperm and egg, chemotaxis is like a barrier. Say how? Say chemotaxis, the word implied chemical attraction. How means a certain chemical compounds which are specific to species will be released. Say fertilizing, a glycoprotein releasing from the cytoplasm of an egg which is specific to that species will be binding with anti-fertilizing, which is an acid protein present in the sperm. This anti-fertilizing of sperm of one particular species will be specific to fertilizing of the ovum of that species, what we call species specific chemotaxis. And after that, the most important, sperm capacitation. Till now the sperm didn't have the ability to fertilize. It should become hyperactivity. That means sperm capacitation means capacity to fertilize. What exactly the interpretation of the word capacitation? It should become hyperactive. Hyperactive means here what is the need for hyperactive? This is very important. The female reproductive tract is like viscous, highly viscous environment. We have discussed about the sperm structure now. The tail or flagellum should be highly active. It should beat like a whip-like structure in a random and erratic manner so as to reach so as to reach the egg. This hyperactivity is essential because it facilitates the sperm oocyte binding and also prevents the polyspermy. So sperm capacitation is very must ensure. After that, acrosome reaction. So these are the 4 steps in present class we are going to discuss. Okay. Then first, the approximation of gametes. Approximation of gametes is the first step. For example, the female reproductive tract will be something like this. Just I'm giving an approximate idea for better understanding. These are the ovaries. No? These are the ovaries. The fallopian tube. No? This is vagina. This vulva. Okay. Here, I told already, the sperms after ejaculation, a million sperms will be loaded in the vagina, and they have to reach the fallopian tube. You know, that means they have to travel a longer distance. I told you, sperm is a very tiny body. It has to travel, swim in a highly viscous environment. Here, that is why the need for sperm capacitation. Hyperactive sperm. The tail should beat the viscous environment so as to reach the fallopian tube appropriately. Here, what we call spot of fertilization is the place or spot of fertilization. So the sperm has to get travel, what we call leader sperm. The leader sperm will be fusing with the oocyte. So it's about the internal fertilization. Okay, if it is external, it is in water. Okay, what we call this is about approximation of gametes. Here it has to locate the female gamete. Okay. Number 2, the second most important, chemotaxis. Chemotaxis we have discussed already. It is a chemical attraction of male gamete. How? There will be a fertilizing and anti-fertilizing, the species-specific interaction. You know, fertilizing and anti-fertilizing reaction. Actually demonstrated by F.R. Lilly. This is fertilizing, and 2, this is anti-fertilizing. This is species specific. Species specific. So as to what is the requirement of species specific? It will avoid the polyspermy. It will facilitate the oocyte binding with, that means sperm binding with the oocyte. So this is very important. So it's just like the sperm will locate easily the egg through this fertilizing anti-fertilizing reaction. F.R. Lilly is the first is a scientist who demonstrated this. Okay. After chemotaxis, the third step is called sperm capacitation. You know, sperm capacitation. Sperm capacitation. Sperm capacity we already discussed. That sperm should become hyperactive. Hyperactive so as to swim thoroughly in the viscous environment. The recent studies suggest that there are certain ionic channels known as cat-spur. Cat-spur ionic channel. Cat-spur ionic channel present in tail or flagellum of sperm. You know, cat-spur ionic channels present in tail or flagellum of the sperm will allow the rapid influx of calcium ions. Now, what makes the sperm highly hyperactive? This is very important. What makes the sperm hyperactive? Hyperactive is because because of the specific cat-spur channels, and this cat-spur channel will allow the influx of calcium ions that leads to formation of flagellum in bends, and this flagellum bends propels the tail with greater force and vigor. That means this greater energy will be liberated so that the sperm will travel swiftly, erotically. That's how they reach the appropriate organ. This is very important. Sperm capacitation will destabilize the sperm membrane so as to make it ready for the acrosome reaction. This is very important. Sperm capacitation that leads to destabilization that leads to acrosomal reaction. Acrosomal reaction. In the present class, we have to discuss at length about acrosomal reaction, because acrosomal reaction facilitates the sperm that who has to fuse with the oocyte. Okay. So the first step is the approximation of gametes so as to locate the female gamete. In case of female protect, this is the process only so as to reach the fertilization. The second step is called chemotaxis, the specific fertilizing anti-fertilizing reaction. The third one is called sperm capacitation. The sperm will develop, that means through series of events, the capacity to fertilize. That means by become hyperactive. How hyperactive means? Here, two means of Caspar channel, the rapid influx of calcium ions makes the flagellum very active. That means so that the cell will erotically swim, and it will facilitate, the egg tries to locate the real place of egg or oocyte. Here the thing is, when it binds to the fertilizing layer, it becomes still hyperactive. That is, this hyperactive nature of sperm is essential. Then the fourth step is called acrosomal reaction. This is very, very important. Acrosomal reaction. Acrosomal reaction. Acrosomal reaction. Before that, we have discussed about certain things connected to this. We knew that in our previous classes we have discussed that spermatogenesis will take place in the seminiferous tubules. What exactly the seminiferous tubule? The wall of the seminiferous tubule is the location of spermatogenesis. After then, they'll get into the lumen of the seminiferous tubules. Then it will reach the epididymis. It will remain for a while. It means what we call storage. Then after that, it will reach the vas deferens or ductus deferens. Then it will reach the spermatic cord. The spermatic cord is the structure that connects the testis to the top of the abdominal cavity. Then it will enter into the pelvic region. Then over the ureter to the prostate. This is a channel, the pathway of sperms before. That means while they ejaculate, this is a pathway of sperms. First, then epididymis, then vas deferens, then spermatic arch, then pelvic region, over the ureter, then to prostate. Here the vas deferens in combination with seminal vesicle forming ejaculatory duct that passes through the prostate and opens into the urethra. Then ultimately it will reach the female reproductive tract. And it's like first the epididymis, then the vas deferens, then it will reach ultimately to spermatic cord, then the pelvic region, then ultimately it will after getting into the pelvic region over the ureter, then over the ureter to prostate. Here the vas deferens plus seminal vesicle form ejaculatory duct that opens into the urethra. Okay, this is how the sperm enters. Then here with respect to acrosomal reaction, the second most important thing before we discuss acrosomal reaction is regarding barriers. Barriers are very important. Oocyte is not a naked structure. It will be, we have discussed in a previous class, it will be surrounded by a 4 layers like corona radiata, zona pellucida, cumulus oophora. But after, that means ovulation, the graphene follicle will be ruptured out. So, but still certain layers are intact surrounding the oocyte. Say particularly the corona radiata, the zona pellucida. The corona, it's like crown. It's like it gives the appearance that as if the oocyte is surrounded by a crown. Then the zona pellucida is very important. It's a thick shell-like structure. It is zona pellucida. Whereas here corona radiata, actually the cumulus cells, they are like gel-like substance which contains, that means it is made up of hyaluronic acid. So it is the first barrier. That means corona radiata is the first most barrier. Then the zona pellucida. Here acrosomal reaction means it has to release certain enzymes. Sperm has to release certain enzymes so that it will what we call enzymatic driven pathway so as to reach the oocyte. Enzymatic means it has to break down the barriers so as to create a tract. Say first break down the corona radiata, then the zona pellucida, then the oocyte. So all these are coming under the head of acrosomal reaction. The enzymatic reaction that leads to creation of a tract or pathway that enables the sperm so as to fuse with that of the oocyte. Last one is called acrosomal reaction. So the first most important barrier is corona radiata. Corona radiata. The second is zona pellucida. Here to counter the same, the sperm will have sperm lysis, what we call sperm lysis. Sperm lysis are in two types, corona penetrating enzyme. Corona penetrating enzyme. And to acrosin or zonalizin. And these two enzymes will break down the corona radiata, the first barrier, then the second barrier, the zona pellucida. Corona radiata actually is a gel-like substance made up of hyaluronic acid. So therefore the corresponding enzyme will be hyaluronidase. This hyaluronidase will break down the hyaluronic acid, but as acrosin zonalizin will break down the zona pellucida. You know, it's a bit complex phenomenon, and most crucial and critical step in entire fertilization process, because it has to penetrate this hardcore shell-like structure, particularly the zona pellucida. Which is nothing but a polymer of glycoprotein, wherein the carbohydrate structures will act as receptors, sperm receptors, like sperm receptor ligand interaction. It is also species specific. It acts like a lock and key mechanism. The sperm in acrosin, the zona pellucida, I will tell you, there will be association between the two during this process, that what we call acrosomal protein with that of that means carbohydrate of zona. This is species specific, like lock and key mechanism or receptor ligand interaction. This is very, very important. What exactly this, how this phenomenon occurs? First the corona radiata will be dissolved by means of hyaluronidase. Then the zona pellucida is the second important barrier. It is very shell-like, a hard shell-like structure, which is a polymer of glycoprotein. The carbohydrate part will act as a receptor, and to this receptor, the acrosomal protein membrane will bind. Just like lock and key mechanism, and is a receptor ligand-like interaction. It is also species specific. Why it is species specific? It will avoid the polyspin. Is very important. At the same time, just like fertilizing and anti-fertilizing chemotaxis, it will avoid the cross reaction. The sperm of different species will not be allowed. Here, what we call molecular level barriers, say chemotaxin and fertilizing and anti-fertilization. Same here, zona pellucida comes under this head. This is species specific, because the carbohydrate structure that of the corresponding partner molecule, what we call acrosomal protein membrane, they'll act like lock and key. That particular species of particular interaction will be allowed here. After this facilitates, it's like lock and key, that means receptor like interaction. It will penetrate through. How? It will release the acrosin or zonalizin. After it will dissolve the corona radiata by means of hyaluronidase. It will break down the hyaluronic acid. The second reaction, then the second barrier, that means zona pellucida, a thick shell-like structure. When it binds, first it has to bind. That means the acrosomal protein and the carbohydrate moiety. Just like receptor ligand, after it binds, it will induce the acrosomal reaction. When it's acrosomal reaction, the acrosomal content will be opened out by exocytosis and vesicularization. It's like releasing the acrosomal content to outside, and so as to release the zonalizin or acrosin will break down. It will destroy that particular zona pellucida pathway, so the sperm can reach and freeze with the egg. Okay, it's about acrosomal reaction that leads to penetration of zona pellucida. It has to penetrate because it is very hard shell-like structure. We have discussed already, because it is made up of glycoprotein. So a specific acrosin, then that break down so as to penetrate the sperm and bind with the oocyte. We will discuss in the form of a diagrammatic presentation for better understanding. How this happens. It's about theory and for schematic representation. In our previous classes, we have discussed that oocytes are covered by different structures. So this is the oocyte, and this is the nucleus, and these are the cortical granules. These are the cortical granules. Then there will be second structure, what we call zona pellucida, zona pellucida. Then it will be surrounding by one more structure called corona radiata. Corona radiata. Corona radiata. It is very important. So the first barrier, corona radiata. Corona radiata. So gel-like substance along with cumulus cells, something like a gel-like substance. Then the zona pellucida. Zona pellucida. Then it is the nucleus. Then these are the cortical granules. Cortical granules. This is how the secondary oocyte looks like. Secondary oocyte. When coming to the reaction, so the millions of sperms will be releasing here, and say one particular sperm will penetrate. First, release two substances, hyaluronidase. Hyaluronidase. Then we'll magnify the same for understanding. Say the acrosomal reaction. What exactly is the acrosome? Say this is after it binds to clear the zona pellucida. This is zona pellucida. So that the content will become out. The inside content will come out. How it comes out? By means of exocytosis. Exocytosis plus vesicularization. Vesicularization. So that the sperm content, that the acrosomal content will be releasing outside and will create a tract. This is the oocyte, okay? To create a tract, okay? This is how, for magnification I am discussing, okay? After clear the corona radiata, what we call the first barrier, the second barrier, what we call the zona pellucida, the green color, okay? This is zona pellucida. That means first it will bind the interaction, what we call lock and key, we have discussed. The acrosomal protein with the carbohydrate moiety of zona pellucida, and what actually the zona pellucida is ZP3 protein, for instance, okay. Then it will exocytosis and vesicularization. It's like it will fusion, exocytosis, that means cell vomiting. Like first a vacuole structure, then cell vomiting, so that the acrosomal content will be releasing outside. One particular structure, okay. And this release content in acrosin. Acrosin, what we call. And this acrosin will penetrate, will enable the sperm, what we call enzymatic penetration, so as to reach this oocyte plasma membrane. This is very important. So the acrosomal reaction consists of two. That means it releases sperm lysins. The first, corona penetrating enzyme, what we call hyaluronidase, it will break down the gel-like substance which is made up of hyaluronic acid. Then the zona pellucida, which it is a hard tail-like structure, it will release, what we call acrosin or zona lysine, so that the sperm will penetrate to zona pellucida and ultimately reach the oocyte. This is essentially important because to reach the oocyte, these two barriers will be created. Okay, this is about acrosomal reaction. Okay, we'll discuss about the next 4 steps in the next class. Okay, thanks for watching.